জি, আমরা এখন লাইভে আছি। ধন্যবাদ সবাইকে আজকে আপনাদের সময়ের জন্য। Welcome everyone to this very special live by Community of Biotechnology in this CRISPR Fest. So this fest actually is one of a kind because in this very moment, Community of Biotechnology has arranged this amazing event where we have been having so many engaging and many interactive sessions. And that's how we're going to be very familiarized with these new concepts which are circulating around. And also, we got many guests there. They are actually working with this particular technology for a while, actually, and they have been contributing to our not only in Bangladesh but also outside of the country and different researches with the very CRISPR technology. So here today, actually, we have very special person, Professor Tofazzal Islam, sir. And actually, if we want to, introduce him, that would be very long. I mean, this is a very long list, the achievement of sir. So still, I'm gonna tell a very small piece of that introduction. Sir, Dr. Muhammad Tofazzal Islam, sir, he's a fellow of Bangladesh Academy of Science, fellow of the Fulbright Commonwealth JSPS and Alexander von Humboldt Foundation. And he's also professor and founder director, Institute of Biotechnology and Genetic Engineering, IBGE, of Bangabandhu Sheikh Mujibur Rahman Agricultural University, BSMRAU, Gazipur, Bangladesh. And now, if I want to talk about the number of publications and how he's involved with research, I'm not going to stop today. So actually, sir will be conducting a session, very fruitful one today. And this will be very interesting on how CRISPR-Cas technology will be used for advancement in agricultural research. And I'm sure, like, if sir wants to add many information, I think a couple of the bits of introduction you can have from his side as well, because I am not enough to convey all the records. So now I'm going to thank Committee of Biotechnology for arranging this event. Committee of Biotechnology is a non-profit organization that has been working on science group and science networking and also scientific enabling other community in this country. And now actually this community has been growing just outside of the country. And many of the bio initiatives, especially bio, but I'm going to say science initiatives, they are also together on this. And in this particular event, this CRISPR Fest, that's actually reached more than 100,000 people. And also, we got response more than 70 countries all over the world. And we're actually thankful to all the participants for your graceful time. And also, I'm thanking again all the organizers and Dr. Professor Tofazzal Islam sir for his graceful presence. এবং আপনাদের সবাইকে শুভেচ্ছা এবং ধন্যবাদ জানাচ্ছি আপনাদের সময়ের জন্য। আজকে ক্রিসমাস ফেস্টের এই বিশেষ আয়োজনে আমাদের সাথে আছেন তোফাজ্জল ইসলাম স্যার, যার পরিচয় কিছুক্ষণ আগে আমরা দিয়েছি এবং ওনার পরিচয় নতুনভাবে আসলে দেওয়ারও কিছু নেই। তবে আজকে কৃষি খাতে ক্রিসপার টেকনোলজির ব্যবহার এবং ওনার নিজস্ব রিসার্চের মাধ্যমে উনি কিভাবে আমাদের রিসার্চ কালচারে এবং আমাদের সরাসরি কিভাবে জিনিসগুলোর মধ্যে উনি অংশগ্রহণ করছেন তার ব্যাপারে একটি বিস্তারিত আলোচনা হবে। এবং আপনারা সবাই প্রশ্ন করতে আপনাদের অনুরোধ জানাচ্ছি। This will be a very interesting and interactive session. Please ask question as much as you can. We will start here and we'll try to contact all of them. Thank you so much. আমি এখন স্যারের কাছে প্ল্যাটফর্মটি হস্তান্তর করছি। ধন্যবাদ সবাইকে।
Excellent. Thank you so much, Salman Promon, for generously introducing me. A good evening. I'm really excited to accept the invitation by community of biotechnology. Actually, this young community of biotechnology is now international, and they are doing outstanding job in culturing the frontier technology to shape our society and progress our civilization. Agriculture, I am an agriculturist by training, and obviously I am a student of biotechnology and genetic engineering. And I welcome from my newly instituted a newly established Institute of Biotechnology and Genetic Engineering to my today's discussion. Actually, the CRISPR-Cas technology is in the history of human. I think in the history of agriculture in last 12,000 years, it is one of the revolutionary technology. We are now living in the CRISPR craze, and I shall show some of the exciting, progress associated with the CRISPR-Cas technology. So I am obviously a fan of this technology, and you can see in the background a book I am editing, the CRISPR-Cas methods. This is the first book the Springer is publishing, and first issue is now online, and second volume is now in the pipeline. I again welcome all of you, and I shall try to show some of the slides, and then we shall discuss a lot. As Salman mentioned, the most important event I am expecting from your side, asking lots of questions, any type of question, because asking question is obviously an expression of intelligence and obviously a big contribution to the scholarly communication. So I am expecting some exciting question, and I shall show some of the slides which describes the illustrate the art of CRISPR-Cas technology, as well as the progresses that we made with this technology, and the progresses that we will make by using this technology, and our involvement with this frontier technology. So I am going to show you some of the slides first and discuss, and later we shall talk more. Let me try. We should involvement with this frontier technology. So I am going to show you some of the slides first and discuss, and later we shall talk. Can you see the slide?
Yes, sir. Yes, sir. We can see the slides.
Excellent. This is the first slide. I'm going to talk on CRISPR-Cas technology for a new revolution in agriculture. You already received several lectures as well as discussion from some big stalwarts who have been working on CRISPR-Cas, and a few minutes ago, Professor Tofazzal Islam, who is an icon in biotechnology in Bangladesh, she also delivered a talk, and I truly enjoy it. CRISPR-Cas technology. You all know that now it is the talk of the world. The Jennifer Doudna and Emmanuel Charpentier, two ladies who discovered this technology, received the Nobel Prize in Chemistry 2020. So let me express my heartiest congratulations to these two winners, and it probably the first case that two ladies together received a Nobel Prize. And they discovered a game-changing, the sharpest, technology, one of the sharpest technologies in biotechnology. As I choose, to discuss on the impact and utilization or application of CRISPR-Cas technology in agriculture, this technology is a robust technology, can be used in bioediting in all branches of biology. I choose agriculture because, food is just like medicine, and food is medicine for every day. And I would like to quote the words of Alfred Nobel. He said, "I would rather take care of the stomachs of all living, then the glory of the departed in the form of monument. That means without food, we cannot survive. The struggle for food in all ages of the civilization existed. So agriculture, we estimate that around 12,000 years we had the long journey and fight against the hunger. So in the all ages of the civilization, ensuring food security was a big challenge. I would like to express my profound, thanks as well as congratulations to late Professor Norman Borlaug, who is known as the father of Green Revolution, because in the history of agriculture, there were several revolutions, but the Green Revolution led by Norman Borlaug, who started from Mexico in mid 20th century, was the most robust revolution in the human history. And what is Green Revolution? Green Revolution refers to a great increase in crop production in the second half of the 20th century through the use of fertilizers, the use of agrochemicals, cultivation of high-yield crop varieties, and mechanization. So till now, we are using these weapons for increasing the crop production. So Green Revolution helped us a lot. And this simple graph, which I prepared based on the available data of human population in the Wikipedia, here you can see after the birth of Christ, the world population was almost static. You may ask why almost static? But after two revolutions made by human, one is industrial revolution, another is green revolution. Now, do you know how much population on this earth? We have now around 7.3 billion people on this earth. But you see, until 17th century, world population was almost static. Due to shortage of food, famine, as well as different diseases and different natural calamities actually controlled the human population. At that time, the famous economist Malthus, he made a law or theory. He proposed that human population is increasing geometrically and food production is increased arithmetically. As a result, human population would be controlled naturally. But after Green Revolution, Malthusian law disproved by Norman Borlaug. So Green Revolution and Industrial Revolution, that means the discovery of engine in Europe, especially in UK, and the use of fossil fuel for running the engine. These two revolutions made human incredibly powerful on this earth. But these two revolutions also brought some of the challenges. For example, Industrial Revolution brought a new problem called climate change. That means global warming. It is one of the most talked issue worldwide. And Green Revolution obviously helped us to, feed billions of people on this earth. But due to the application of agrochemicals and other, high inputs in agriculture, it deteriorated the natural resources, biodiversity, even soil health. So these two revolutions we found now also associated with lots of big problems that needs to be addressed by using the new technology. For example, in case of Green Revolution, in last 4 decades in Bangladesh, we increased our food production, especially the grain production, 4 times. But if you see last 4 years or 5 years, you can see they are nearly in plateau. The food production are not increasing as they increased before. But our population is increasing faster. So now the question is, we need why we need a new green revolution in agriculture? Actually, we need a new green revolution in agriculture. Let me tell you some point. New green revolution obviously associated with some new change. And changes in the human history due to innovative minds of human and their research, the research and seeking new knowledge. Always we are changing. So change. Norman Borlaug, the father of Green Revolution, who won the Nobel Prize for peace, he said, "Change we must, or we will perish as the species, just as did the dinosaurs in the late Cretaceous." So let me show you some of the real statistics in this world. By the year 2050, the world population would be nearly 10 billion. Nearly 10 billion. But per person, land would be 0.2 hectare. So land is shrinking. Agricultural crop croplands, cultivable lands are shrinking, and population is increasing. So what we need to do, we need to obviously increase the productivity. We obviously need to increase the quality of the product by using new technology. Here you can see, can we? The question is, can we produce sufficient food from 2.2 hectares of land per person for feeding the future population in this earth? Obviously, it is a formidable challenge because land is shrinking, and new problem I mentioned, the climate change. Climate change is posing a serious threat to the food productivity. The 17 goals set by the United Nations that are known as Sustainable Development Goals, which we have to achieve by the year 2030. The number one goal is end of poverty for all. Second one, freedom from hunger. That means now we have 7.3 billion people on the earth. 2 billion people, around 2 billion people, they are going to bed with hungry stomach. So we cannot imagine a peaceful earth if people go bed with hungry stomach. Another target is sustainable, ecosystems, sustainable consumption and production. So all these four goals, major four goals, are associated with agriculture, because lots of people all over the world, their livelihood is associated with agriculture. And we can make lots of technology, lots of advancement, but we cannot make a single grain in the factory. Without agriculture, we cannot ensure our food. So question still, I want to explain a bit why food security is a challenging task. To meet the growing demand by 2050, a global increase in crop production of 70 to 100% is required to feed the 10 billion people. Climate change poses an increasing risk to crop production, and climate change is posing serious threat to crop production through higher incidence of the pests and diseases, as well as causing abiotic stresses like heat stress, salinity problem, and drought problem. Lots of new problems associated with the climate change, and that are clearly big threat for the food production. The rate of increase of yields brought about by the last green revolution has been steadily declining. This is a very big concern issue. That means the inputs or technologies developed by the green revolution, last green revolution, they are not working as they worked before for increasing the production of the crop. The conventional strategies used for crop breeding are laborious, time-consuming, and complicated. That means we need something smart, faster, and time-saving. So more effective and time-saving breeding methods are required to satisfy the growing demand of food for ensuring the food and nutritional security of the future population. Now the question is CRISPR. CRISPR, yes. What is CRISPR? Already from many lectures, you already know that the word CRISPR, that is the abbreviation CRISPR, stands for Clustered Regularly Interspaced Short Palindromic Repeats. And you already know what is it. It is the sharpest genome editing technology that targets a specific section of DNA, makes a precise cut or break at the target site, and can do one of the two things. What are those? First, makes a gene non-functional. If it is a bad gene, then if you can make it non-functional, then you can improve the trait of the crop. For example, a susceptible, disease-susceptible gene, which we call S gene, a negative regulator of the disease. If we can make it non-functional, then pathogen exists in the environment but cannot recognize the host plant and cannot cause the disease. And second point is replace one version of a gene with other. Not only these two, CRISPR can do lots of much more things. Professor Jeba Islam Shiraz already discussed lots of things. CRISPR can make biodiversity, genetic diversity in the crop. CRISPR even can do some magic, like wild crops. Wild crops, you can make them domesticated, because last 12 years we only domesticated 15 to 20 species of the plant, although in the earth 400,000 species of the food crop are available, but we only focused 15 to 20 species. So they are susceptible to lots of big problems, but there are lots of wild relatives which we call orphan crop or neglected crop species. We can make them domesticated within few generations. So I shall discuss some of the exciting things associated with the CRISPR. Already you know that what is gene editing or genome editing. Professor David Baltimore, who got Nobel Prize in 1975, he stated that precisely altering genetic sequences in living cells, including those of human, at much accuracy and efficiency than ever before, he defined as genome editing or gene editing. And I can simply say any techniques using site-directed mutagenesis to make a small change to DNA sequence at a known locations is called gene editing or genome editing. The entire genomic material of an organism is called genome. So DNA, RNA are the nucleic acids, and they are the black box where all the features of an organism are written in secret code. So if you can change this secret code by using any technique, you can change the properties of an organism, even in plant or even in human. So CRISPR technology, already you know that it is actually discovered through fundamental research. In 1987, Professor Ishino in Japan, he found that when he sequenced a bacterium, he found that some clustered some unusual sequences repeatedly present in the DNA sequence of a bacterium. And from 1987 to 2012, that means 2012, scientists did lots of basic research to discover the mystery of the presence of the CRISPR, later it is known as CRISPR, why present in the DNA of bacteria and archaea, the prokaryotes. And over the periods of time from 1987 to 2012, scientists discovered that it is nothing but the adaptive immune system of bacteria. In our case, we have antibody and we have other molecular weapons to tackle any, pathogen enters into our body. But in case of bacteria are prone to infected by the virus. So bacteria, virus, they cannot replicate themselves. They use the transcription and translation machinery of the host, like COVID-19 virus. It cannot make its own copies outside the human host. So what it does, it enter into the human body and then use the human, nucleic acid copying mechanism for making its own copy. Similarly, a virus infect the bacteria or archaea and then use the transcription and translation machinery to make its own copy. But through the evolutionary process, bacteria somehow they cut the small part, those who survived during the the infection by the virus, they some get the segment of DNA from the virus, invading virus, and they preserve there, and there are associated the protein. So when CRISPR RNA express this associated protein, can easily remember contains the memory of the that segment of the invading virus. And then what they do, they can cut the invading virus and tackle the virus for their infection. That means using their own machinery for the copying. So it is an immune system of bacteria and virus. And until 2012, all scientists who published hundreds of fascinating papers on through the basic research, they discovered this mechanism. That is, bacteria by using CRISPR-Cas system can cut the invading viral DNA and make a virus inactive and cannot allow to use their copying system of the nucleic acid. So it was up to that story, basic, research and fundamental understanding of the mystery of CRISPR. But in 2012, a game-changing paper published by Emmanuel Charpentier and Jennifer Doudna. These two ladies actually in 2011 they met together in Puerto Rico, a conference, and they discussed how can they collaborate with each other, and they within years of their collaborative work they published this this paper, that is a programmable dual RNA guided DNA endonuclease in adaptive bacterial immunity. As you know, it is a CRISPR-Cas system is an adaptive bacterial immunity. But what they change here and how they made the game-changing technology, they thought that if we can use this molecular scissor for cutting any other DNA not inside the bacteria, for purposively to edit the to change the DNA of in a specific location or a specific gene, then we can do the engineering very smart way. Actually, they were very successful, and they developed this method. This method, obviously, you received that many lectures that by using a guide RNA, you can design the guide RNA complementary to the targeted DNA where you want to cut, and you make a complex with Cas9, and then it can cut the double-stranded DNA. And here, Cas9 or Cas12 or other, there are several other enzyme or protein have already been discovered in bacteria, and you can cut the double strand of DNA in a specific location very precisely and very quickly. And after cutting the DNA, DNA has own repair mechanism. So if anyhow a DNA is damaged, immediately it repairs. And it repairs in two ways. One is non-homologous and joining repair pathway. In that case, what can it do? The targeted, the nucleotide, a single nucleotide may be damaged and it is repaired. That means one indel mutation taken place. And due to lack of one, nucleotide in a specific gene, it may be non-functional. And you can even delete a specific gene in the sequence, because if you cut in two places and remove a gene, you can do that. And you can also insert a gene. If you put a template gene which you want to insert in the space, you can do it. Another pathway is HDR, homology directed regulatory pathway. And here also homology, for example, double stranded, if you put the homologous end of your the donor DNA in the medium, after cutting, it can paste it like this way. And here you can change, add new gene, or you can rearrange the sequence of the DNA, and you can get gene insertion or replacement, or even you can correct DNA. Because if it is a sequence is wrong and undesirable, you can correct it and homology directed, because end is homology directed, so it would easily fill up the gap and gene correction can be taken place. So this is very simplified way I tried to show you, but it is really work like this way in a magic. So I can show you here what is the components for gene editing. You need a single guide RNA. You can design it, and synthetically you can do it. And Cas9 nucleus. Nucleus means which can cut the nucleus. So you can combine two and makes the complex, and it is the Cas9 complex with guide RNA. How it would do? For example, you targeted this sequence and you design the guide RNA in a like that way. For two, spacer adjacent motif is required for such targeting. Already Professor Islam Shiraz and Professor Jakir described very nicely. 3 nucleotide this is, sequence is required, and it is present in the all living organisms. So what would happen? Here you see it would come here and find the complementary sequence and make the double strand break like this, and then through own DNA's own repairing mechanism, you see it can make a change in the DNA sequence. Here you see 3 nucleotide is changed. So this is very simple process and has been used in almost all eukaryotes and prokaryotes for changing the DNA. And lots of news after 2012, the discovery of that genome editing. It became a very hot topic in the scientific community, as well as in the intellectual community. And in 2012, CRISPR-Cas technology have successfully been used to edit the genome of plant by 3 groups, and these 3 papers published in Nature Biotechnology. One of the pioneer of plant genome editing was Professor Sufyan Kamaun, a team leader of a research group in the Sainsbury Laboratory, UK. And I am showing him because he is now one of our collaborators, an amazing scientist. You can browse Sufyan Kamaun in the internet. You can get lots of information. Diversities in genome editing by CRISPR-Cas technology. After the discovery of genome editing by the guide RNA by Emmanuel Charpentier and Jennifer Doudna's collaboration, lots of advancement in CRISPR-Cas technology have been done. Now we can do lots of more things. Here you can see, as I mentioned, even single indel can be done by using single guide RNA through the homologous and non-homologous end joining. You can use, double a guide RNA, and you can target a specific gene to excise from the DNA. So a chromosomal fragment even you can remove from the DNA of an organism. And you can even use a donor. If you put a donor fragment, you can add a gene, insert a gene, a fragment in the non-homologous end joining. And you can donor repair template. For example, you can repair if there is something wrong in the sequence, and if it is desirable, you can do it. And even you can do the silencing via Cas9, that is RNA. In the RNA, even you can remove the silencing via RNA processing Cas9 for specific sequences of mRNA leading to translational sequencing. So even in RNA, you can do the editing and which impact on the translation process. That means protein synthesis. And recently, more precise advancement has been taken place. For example, if you want to target a specific base for editing, you can do it. For example, in case of Cas9, cytidine deaminase, if you make a complex and then you can target, this CG, you can change into TA. And similarly, adenosine deaminase enzyme, if you fuse with the Cas enzyme or deaminase fusion, you can make lots of other changes. For example, guanine cytosine you can make from adenine thymine pair. And similarly, even two base you can make editing. So by base editing is another very, very interesting, and by using base editing you can make the change in the phenotype. So there are lots of other changing you can do by using prime editing. So more precisely, you can without double strand break, only single strand break, you can cut the DNA and make the changes. For example, prime editing is the latest one, and you can do more precisely and more easy way, more conveniently, just cutting a single strand and make the changes and edit the desired gene or genome. So stacking beneficial alleles, you can do stacking beneficial alleles by using homology directed recombination. So for example, beneficial alleles you can stack in the targeted chromosome. So then you can make a huge impact on the your target plant. CRISPR delivery, as you know, that it is really one of the challenging issue. How can we you deliver CRISPR-Cas reagent into the plant cell? Because plant cell has a thick cellulose containing very thick cell wall. So there are several methods like biolistic bombardment method, nanoparticles, agrobacterium tumefaciens mediated method, or viral RNA you can use to deliver the CRISPR Cas system into the plant cell for the desired editing. So lots of new advancement have been done within the last few years, and if you check the literature, you can see every week hundreds of new papers are publishing, and CRISPR is becoming a powerful user-friendly tools for the researchers. And what I can say that in future you can see a CRISPR Cas method like PCR in all biology lab, as it is becoming a user-friendly like your smartphone. And when I started to work on CRISPR-Cas for editing the genome of wheat blast, I went through lots of scientific paper. In the scientific paper, the methods, results and methods, materials and methods part in the big journals, always you have to write very briefly. So essential, if any step is missing, you cannot use it in the laboratory. This is why I decided to publish a book, a handbook, for which is called a protocols book, because in different organisms, different plants, although principle is same, but there are slight changes in CRISPR for efficient use of CRISPR-Cas methods. So I decided to publish a book, and this book is available, CRISPR-Cas methods. So you can find it in the internet, and the second volume is now under process of preparation. And I told you the story, CRISPR story started from 1987, a strange phenomena observed by Ishino group, and in 2020 it received the Nobel Prize in chemistry. Really very fast from the first discovery to the translation of the basic research into a technology. It took only a few years. For example, in 2012, CRISPR-Cas developed as gene editing tool. So only 8 years it took to receive the world highest recognition, that is Nobel Prize in chemistry. What is the unique in CRISPR-Cas technology? It is derived from a natural process found in bacteria to protect themselves from pathogens, that is virus and plasmid, targets genes for editing or regulating. Regulating means you can rearrange, you can insert, you can delete, you can make a single base editing, even you can do epigenetic changes. And it is comparable to Photoshop. So in the textbook, in the for example, in a book, a manuscript in the Microsoft Word, we can edit as we like. In the Photoshop, also we can edit any picture as we like. In the CRISPR also, you have enormous opportunity to edit the DNA and RNA. Now, question is why CRISPR-Cas technology is superior? Because already you know that there were several gene editing technology discovered before the CRISPR-Cas. For example, zinc finger nuclease or transcription activator like endonuclease. So they also can cut the DNA, but they were very cumbersome and very, very complicated, very expensive, requires sophisticated protein digestion, protein synthesis and validation. But in case of CRISPR, only guide RNA design, and even lots of companies are now offering, if you have a specific target, they can design the guide RNA. You can browse in internet, you can see, you can get the all, how to say, toolkits. CRISPR-Cas is now a toolkits. You can get the toolkits from the market, and you can do your job. So CRISPR-Cas is cheap, relies on single guide RNA to target site specificity, can be readily designed based on genome of choices and in multiplexed fashion. This is very important. At a time, in a different location, you can make editing. But in case of zinc finger and talent, it was very, very complicated. And several genomic loci, you can make perturbation. System create stable and heritable mutation. That means once you edit it and develop a new variety of plant, it is heritable and it is stable. And another fascinating thing is that transgene-free plants in a few generations. Because breeding of a any new disease resistance or any other trait requires 10 to 15 years, and lots of as it is not specific, lots of other things came and very cumbersome, time consuming, but within a few generations you can get transgene free plants. In case of recombinant DNA technology, antibiotic resistant genes and other transgenes create serious problem for biosafety issues and other things. So edited plants variety can be used for plant breeding because you can use make a diversity, genetic diversity, by chopping in the DNA in different location and make the genetic diversity, and that could be used for plant breeding as new genetic material. New set of CRISPR-Cas systems such as Cas12 or Cas13a, they are really much more convenient than Cas9. So future of CRISPR already described. It can be used in human gene therapy, agriculture, crops, and animals gene editing, synthetic biology, pathway engineering you can do to make new drug, new biomolecules, and programmable RNA targeting, screens for drug target ID. Yes, you can use specific DNA of the pathogen, and easily you can detect the pathogen very fastly. For example, COVID-19, Sherlock method by using CRISPR-Cas Feng-Jiang developed a smart technique to first detect the COVID-19. Ecological vector control, mosquito sterilization, viral gene disruption, pathogen gene disruption, and most important is in basic biology, CRISPR-Cas is now a very powerful tool at the point of genomic and post-genomic era to understand the mystery of life. So you can rewrite the life by using the CRISPR-Cas method. Now, I would like to share some of the success stories of genome editing. For example, mildew is a dangerous disease in many crops, and mildew resistance locus, MLO, have been discovered by scientists, and it is a classical S gene. That means negative regulator of the disease. It helps the pathogen for infecting the plant. So presence of this locus is associated with the susceptibility of the disease, and the fungal disease like Plumeria graminis in barley. The gene was mapped and occurs in small to medium-sized families in the genome of higher plants. Many higher plants contain the genome, and the protein is structurally related to G protein couple receptor in metazoans, and scientists targeted that genome. Here you can see this is the protein. It is calmodulin binding protein, MLO, and MLO found powdery mildew interactions in many plants, and scientists designed the CRISPR and used it for deleting the gene. And what happened here you can see in case of tomato, the targeted here you can see plant 2, plant 8, and plant 10. This part of the sequence is deleted. And what deleting resulted in case of wild type, the normal one, you can see the disease symptom. But after deleting of that S gene, MLO locus, you see an amazing result. That means no disease, and plant became immune. And scientists checked whether any Cas9 or any other trace of tDNA or any other trace of the transgene present in the edited plant. They found nothing. Here you see no tDNA has been found in the transformed plant. After Illumina sequencing, it has been confirmed. What does it mean? This edited plant only less these few nucleotides. And 48 to 49 base pairs are deleted. That means from an ocean, you took a few drops of water, and it is naturally, always in the plant, it is happening, such type of mutation. So this type of mutation, less DNA, is no problem associated with the, biosafety and biohazard. If it is not affecting the yield of the plant. But so it is very nice. By using, base editing, lots of new improvement has been developed. For example, I told you base editing method, for example, cited in deaminase domain, and single base editing or double base editing. Lots of improvement in, for example, rice, our major crop, have been developed. For example, nutritional improvement, herbicide resistance, enhanced nitrogen use efficiency, because nitrogen use is one of the important costly input in case of rice production, regulates cell senescence and death. So lots of things, amazing things are coming, just deleting, making a one or two, base editing, you can get such a robust impact in the plant. And one of the most fascinating story I am showing you by this, image. Here you can see domestication, acceleration of the domestication of wild tomatoes. Wild tomato still exist in the nature, and we have now the modern tomato. Modern tomato, number of fruits, very high number. They are flowering higher, and taste is also good, and vitamin C level also higher. So over 10,000 years it took to get evolution, or through the breeding process we got the domesticated the tomato. But scientists discovered what are the after sequencing, they discovered that only few genes over the years were changed from the wild tomato. So by using CRISPR-Cas technique, they targeted all these genes to change in the wild tomato, and they made the change. And they found that amazingly de novo domesticated tomato, just like our natural one. But in addition, in wild tomato, they have the high disease resistance as well as tolerance to abiotic stresses. For example, salinity tolerance. So these two characters retained, and we added the domesticated genes into the wild tomato, and we got the scientists got these amazing changes. That means you can make domestication of wild relatives or neglected plant if you know the domestication genes. As now genomic technology have been advanced tremendously, so most of the 15 to 20 species of our domesticated food crops which we have been used, they are unfit for the climate change adaptation. You can now target the domestication of the wild relatives, and it is an amazing success, and it would make a new revolution in agriculture to fight against the climate change. So you can ask how many genes have been already edited. Almost all economically important plants, scientists tested CRISPR-Cas gene editing system, and they successfully did the job, and resistance against the pathogen, insects, as well as resistance against the abiotic stresses like salinity, like drought, like cold. Tolerant plants have been developed. That means it works for making a desirable change and making a climate smart crop for the future, for ensuring the food security. You may ask me a question whether in the real field any CRISPR-Cas edited organisms are cultivating or used in the practical field. Yes, in case of soybean, disruption of DRB2A and DRB2B genes, double-stranded RNA binding proteins 2 genes for drought and salt tolerance. And as there were disruption of the gene, no transgene was present. USDA in USA, they gave the green pass. No need long course of biosafety. So they are now cultivating in the USA since 2017. Similarly, camelina, which is an oil containing crop, so increased oil content is achieved by editing an undisclosed gene, because a company they didn't disclose it, but it is already got the green pass. Similarly, Citaria viridis, green bistal grass, also released in the practical field. Waxy corn by DuPont, already cultivated in the USA. White button mushroom, a professor in Pennsylvania State University, Professor Young, I met with him, and if you cut this mushroom, they become browning. So anti-browning mushroom, by knocking out a gene, they developed, and it is very fascinating and now free for cultivation in USA. So these are some examples, and new things are coming out, and CRISPR-Cas technique in different countries are regulated differently. I shall tell you later on. USA, Canada, Japan, they consider that CRISPR-Cas edited mutant are not carrying the transgene, and they give the green pass without biosafety. They can go to the practical field. And now I would like to tell some of our story. In 2016, a dangerous disease called wheat blast emerged in Bangladesh and devastated. For the first time, it emerged in Bangladesh and devastated 15,000 hectares of cultivated wheat in 8 districts, and yield loss was up to 100%. And as it was not a familiar disease and totally unknown disease in Bangladesh, the government decided to burn the crop to reduce the inoculum potential of that fungus. And as I am a student of biotechnology and genetic engineering, I decided to understand what is the enemy. And by using field pathogenomics and through a big international collaboration, 31 people engaged from 4 continents, we discovered that the wheat blast in Bangladesh is caused by a South American lineage of Magnaporte oryzae. And it was a big news in our local newspaper, as well as in the big news in Nature, as well as Science. And after this discovery, that is, the pathogen is a fungus, and it is a devastating fungus, can cause 100% yield loss. And wheat is our second food crop. Our next target was to develop a blast-resistant wheat variety. So it was a big challenge, because wheat is a hexaploid crop, and every gene in the genome has 6 copies. So we targeted, as you know, the tomato S gene editing showed the powdery mildew resistance against the fungus. We decided to edit some S genes in the genome of wheat, and we to develop a durable blast resistant variety, because in the world there are no bona fide blast-resistant variety against the wheat blast. And I collaborated with the Sufyan Kamaun. Sufyan Kamaun, actually, he joined with me in determining the genetic identity and origin of the wheat blast, and he is a Royal Society Fellow, and Nicholas Talbot, another Royal Society Fellow in UK, and they joined with me to fight against the wheat blast. And how? By using the genome editing technology. As Sufyan is one of the pioneer of using CRISPR-Cas system in plant, we invited him to Bangladesh, and we trained around 50 people on CRISPR-Cas genome editing technology at our university in the Institute of Biotechnology and Genetic Engineering at Bangabandhu Sheikh Mujibur Rahman Agriculture University. What we did, we targeted, as I mentioned, 10 genes, and by using protoplast, Professor Tofazzal Islam Shiraz told that protoplast is easy for delivery of the CRISPR-Cas system, because cell wall is a very big problem. So in collaboration with Sufyan Kamaun and Nicholas Talbot, we were as well as Pankaj Bhaumik from NRC. So through an international collaboration, we were able to edit 10 S genes in the wheat genome. And we got the wheat edited wheat, and we are testing them now whether they are blast resistant or not. And our next target is, if this concept is proved, that means we can edit the wheat genome successfully. But it is still question whether the transformants, that is edited plants, are resistant or not. Till now, we got only moderately resistant, not like the tomato, mildew-resistant tomato or like that. But as use this construct, this construct is available now in AdGene publicly, and we use the open science and open data sharing approaches so that anybody from anywhere in this earth can join this project. And as this is a novel idea, innovative idea for a developing country to address a real-life serious problem associated with the food security, we participated in a competition announced by Islamic Development Bank, and our idea became the champion, and we got the award in 2018. So this is a running project that is a development of new blast-resistant wheat variety to fight against the wheat blast. Wheat blast has been in South America, and it is a big problem in South America since 1985, but other continents are free. In 2016, it came to Bangladesh, and India is the second largest wheat producing country. China is world #1 wheat producing country, as it is seed-borne as well as airborne. There is high chance of transmission of these diseases in other country and can make a catastrophic problem for the world food security. And last year it moved to Africa, so it is danger on the move. But one big problem is that, for example, in case of there are no a diagnostic tool to monitor the wheat blast fungus, because wheat blast fungus is Magnaporte oryzae. Triticum petotype is specific for wheat. It never infects the rice. Rice one is Magnaporte oryzae petotype. So to detect precisely the Magnaporte oryzae triticum, we decided to develop a convenient rapid diagnostic tool for monitoring and surveillance, as well as to use it in the quarantine. Wheat is #1 food crop for export and import. So during export and import, there is no tool for the diagnosis of the wheat blast in the quarantine system of any country. So we decided to develop a diagnostic tool. And what we did, we used Cas12 enzyme and guide RNA, as Cas12 enzyme can cut if we design a guide RNA, specific location of the DNA, and can cut the DNA and make it inactive. So we thought that this revolutionary technique could be useful. And what we did, we first used, rice and wheat blast, all blast genome sequences. We searched to find unique sequence in the wheat blast. We found MOT 6089, MOT 6099. One encodes a putative acid trihalase protein, another encodes a DAF 341 family protein. And these two sequences have not been found in any other fungi in this earth. So they are very specific. We designed the primer, and then by using Cas12 enzyme, by using primer, we can very precisely detect the wheat blast in the plant as well as in the grain. And we published this paper in a very high-ranked journal recently during the coronavirus engineering, which has a high impact factor. And we, as I mentioned, PCR is not possible to use by the quarantine as well as by the farmers. So to make the method very easy, we used, the strip, paper strip, like a pregnancy, test strip by using Cas12 enzyme and designed guide RNA. Now it is like that. Use of Cas12a for rapid detection of wheat blast specific sequences. So what you do in the buffer solution, you put the sample in the seed or plant material you suspect for wheat blast infected. So if it is positive, the specific DNA of the fungus, Cas12 enzyme, as we designed the guide RNA, it target the that DNA and cut it, and you would not see any bend. But in case of if two bends are present, that means our design guide RNA didn't find the complementary sequence. So it is the smartest technology in the field of agriculture, just like a pregnancy test. And we are now discussing with a US Bangladesh joint company to make it available for the farmers, for the extension workers, for the quarantine people. So by using this technique, within 30 minutes, you can detect the wheat blast fungus in the sample, and it would reduce the fungicide use, because since 2016 in Bangladesh, all the wheat seeds are treated by the fungicide, and it is very expensive. In the field, we are using fungicide, even there is no fungus, because it is symptomless at the young age. But now you can check whether your field is contaminated with fungus or seed lot is contaminated or not. So it would have versatile use and huge economic impact in agriculture. So CRISPR-Cas, I am now at the end of my lecture. CRISPR-Cas technology and GM regulation is very important topic, because as this technology is smart, we are talking that no transgene is present there. But in many countries in this world, there is no law for releasing them fastly. Although USA, Japan is very conservative country, but they gave the green pass, and Canada and some countries are already gave the green pass. But European Union, they are reluctant. They, in Europe, you have to follow the biosafety guidelines. But in Bangladesh, we have no law. We are advocating for generating a law. Because Bangladesh is a country where food security is a very big challenge. So we need a revolutionary technology for enhancing our food production and ensuring food and nutritional security. So plant products from mutagenesis specify exempted from European GM regulation. Mutagenesis, if you do mutagenesis by using chemical mutagen or gamma radiation, it is acceptable. But if you use CRISPR, then it is not acceptable in Europe. In many country crops, cultivars from mutagenesis, wheat, rice, apple, ornamentals, they are now cultivated worldwide. transgene-free CRISPR-edited crops are outside of GM regulation in Japan, US, and Canada, I mentioned. In US, CRISPR-Cas products outside of regulation by USDA. So CRISPR-Cas like amazing technology is necessary for making a new green revolution, as lots of new challenges are coming due to the global climate change, like new infestation of the disease like wheat blast, new problems like drought, stress, salinity, and a large part of our South Bengal is affected by the salinity. Already Professor Tofazzal Islam Shiraz mentioned. So we need revolutionary technique and to adapt to the changing climate. I would like to quote our great poet Rabindranath Tagore. You cannot cross the sea merely by standing and staring at the water. You must use revolutionary technology to make new revolution, and science and technology giving the solution of any problem exist in the society. So concluding remark is the world population is expected to increase around 10 billion by the year 2050. The CRISPR-Cas system became the gold standard method for breeding smart crop for resiliency in climate change. The CRISPR-Cas toolkit is simple, efficient, and versatile revolutionary technology for investigating plant biology, precision breeding, and creating genetic diversity in plant. We foresee the application of CRISPR-Cas technology in various crops, revolutionize agriculture in a green revolution to ensure food and nutritional security of the ever-increasing world population. I would like to share with you that I received a chapter recently from an US scientist that they had a very big project to introduce CRISPR-Cas, technique to train the high school and college students, and he made a very nice package, to do in vitro test of the, gene cutting and engineering, and you could find in the volume 2. And I am thinking to introduce such thing in Bangladesh. So most important is our university curricula, as well as high school or college curricula needs to introduce this revolutionary technique, so that our young generation can be prepared in a way so they can lead our nation as a food secured, nutritionally secured, a developed country by the year 2041. I am very much thankful to the community of biotechnology, and I'm very much thankful to lots of international collaborators and donors. And I welcome all of you who are interested to do, to use frontiers technology for any research, or to learn new techniques, to do interning. You are welcome to my Institute of Biotechnology and Genetic Engineering. We have, fortunately, fantastic facilities as well as funding for supporting the young people. Thank you so much.
Thank you so much, sir. That was amazing, informative, and that was interesting as well. So we are having so many questions. Before jumping into that, actually, I want to add that sir here, he has been supporting this young community of scientists for a long time. I mean, as you can see, he has been like working with so many other countries, many collaborations. I'm not sure if he sleeps or not, but he has been working, he's been giving time, not only his research, but also people like us, the students, everyone. Actually, he has time for all of them. I'm not sure how he's managing that, but he's trying to make out the best out of it. So thank you so much, sir, for being such an inspirational character, and
It's
also
my
like supporting
pleasure.
us. Thank you so much, sir. So, sir, can you like allow us to take questions so that like we can
Of
come up
course,
with
of course.
Great, excellent. Great. Yeah. So the first question arrived.
Do eye cell cardiomyocytes proliferate after plating? Can we make our own cryopreserved stock? I have no experience on this, cell. Because I am, basically, I have been working on microorganism and plants, but you may check it in the net. Should be, preserved, because all types of cell line we can preserve, and there are you should have certain, protocol to follow before, keeping in minus 160 or like that. Thank you. Can single guide RNAs used for CRISPR editing be used for adenine base editing? Yes. In that case, I told you an additional enzyme needs to be fused to the CRISPR-Cas system, and single base editing is possible. Even two base editing also can be possible. And single base editing technique or double base editing technique have been used to edit the rice genome to improve the plant with lots of new traits like nitrogen use efficiency. Why do I get a lot of colonies upon transformation of digested lentiguide puro? This is a tough question. Lentiguide puro, I am not sure why you get lots of colonies. I'm not sure whether it is associated with the contamination or it is the natural phenomena. I'm really sorry because I'm not experienced on this. Is there any aspect that CRISPR system may outcompete CRISPR? Salman, in the previous session, he told the new technology always, surprise us. CRISPR should not be the ending. CRISPR is a surprise at this moment. Maybe CRISPR leads to new surprise. And obviously, CRISPR is now the sharpest and the smartest and precise and faster technique for gene editing compared to any other existing technologies. But who knows, one day you can surprise the whole earth making much more surprising technologies. CRISPR has lots of still limitation. For example, off target, as well as complicacy in transformation, because all plants are not easy to transform. So lots of challenges are there. In future, you may see simply spraying something, you can edit the gene, and it is coming. And in case of CRISPR also, such amazing improvement is going on. And as CRISPR is very, very useful technique for basic research, I'm sure new technology would come which would be much more sharper than the CRISPR. Thank you. Starting with genome analysis, what software to use? Genome analysis, actually lots of software, which should I tell you? So please, there are lots of open source softwares also. So please browse the internet, and which organism you are going to analyze is very important. Genome of human or genome of bacteria or genome of plant. So depending on the size of the genome, you can find suitable analytical tool, bioinformatic tools in the internet, as well as community of biotechnology always organize bioinformatics training, and it is an important area for offer the training to the young people. And I appreciate the community of biotechnology and other young people who are doing fantastic job, and it made us proud, because I checked PubMed and found more than 150 papers published on COVID-19 by our young researchers in world-leading journal, simply doing the bioinformatic analysis. Thank you.
So some of them are actually making questions right now. So that's the information.
Yes, can I pull wild type and CRISPR-Cas cause cells in the same RNA sequence sample and demultiplex them by puromycin resistance cassette expression? Yes, it is possible. It is theoretically it is possible, but I'm not sure the evidence is already been established or not. But theoretically, it should be possible. Nairita Hassan Faruqi, the one of the organizer. Thank you very much for nice question. What are the ways to inactivate a specific gene in a cell? Yes, you can make inactivate a cell by using non-homologous end joining, because if you can target that gene by specific guide RNA and then make a double strand break in the middle of the gene, and then during DNA repair, natural repair mechanism, an indel or mutation will be happen, and it may result non-functional of the gene. And even you can remove the whole gene or a fragment of the gene by using a CRISPR-Cas method, and in any cell it can be used because it has been already tested in most all eukaryotic and prokaryotic organisms. Next, yes, Shagorika Shahriar, can you please explain the phenotypes differences of CRISPR-Cas9 edited cell model? Yes, phenotype differences. For example, if you want to change a flower color, if you edit a gene responsible for the pink color, and if it deleting make it white, so at the flowering stage you can see it. We can check it by using PCR also, by using the sequencing also. But for example, phenotype change, Professor Jeba Islam Shiraz described the salinity tolerance. If you edit a gene associated with salinity susceptibility gene, if you edit and make it non-functional, and after editing, when you get the seedling through the tissue culture, you can check them in different concentration of salinity, and the edited one, right one, would show the tolerance, can grow well on the saline water or saline medium, but non-edited one can't. So you can easily get the phenotypic differences. A non-edited one will die in the higher saline condition. Thank you so much. Ghulam Mahbub Faisal, can we use CRISPR-Cas to increase livestock quantitative traits like milk and meat production? Very big question, because some of the characters are polygenic. So in that case, we need to have thorough knowledge which genes are involved in regulating a particular trait. If you know that precisely, then you can design a multiplex CRISPR-Cas editing system, and then you can do it, as you have seen in case of tomato, domestication of tomato. Scientists in 2018, they were successful. Within 2 generations, they domesticated a wild one, just like which happened by 10,000 years or like that. So amazing. So in case of wheat and meat production, milk and meat production, I am sure. And in USA, in California, they used CRISPR-Cas in animal for changing many traits, including the meat production also. Please browse the net, and even in the YouTube, if you browse your keywords, you can find lots of lectures. CRISPR is really amazing. No technology is making such a robust impact to the society. So CRISPR is like that, shaking the all nerves of human civilization. Thank you. Maisha Farjana. In case of RNP-based CRISPR, is a single guide RNA scaffold universal? Single guide RNA scaffold not universal. It depends on the organism also, but in commonly in some cases it can be used. Nairita Hassan Faruqi again. What other options are available for validation of genome-wide association studies candidate genes apart from RNA and CRISPR? This is a big question. Option is limited by sequencing, and RNA obviously very interesting, but CRISPR is the best. Other options I don't know, but nowadays by using different bioinformatic tools, genome-wide association is an important area for research in plant science. Shagorika Shariar, what's the most efficient CRISPR-Cas9 method for transgene, example protein sequence, to be integrated in CHO genome with stable expression level? Most efficient CRISPR-Cas method for transgene to be HDR, homology directed recombination. Although it has lots of problem in the early stage, but recent prime editing and other advancement, helping and making it more efficient for integrating a transgene in any genome. Fahad Alam Said, researchers have been found where the CRISPR spacers can prevent the lysis process in virus. Could you give insights to how it affects the viral genome? Yes, this is interesting. In viral genome, CRISPR, if CRISPR sequence is complementary to the viral genome, if you naturally in bacteria, they have the memory there, so invading virus they can target and inactivate. But in case of COVID-19 and other virus, we can design the guide RNA depending on the target sequence, unique sequence of the specific virus, and like Sherlock method in the earlier section, one student asked a question in case of COVID-19. That method seemed very efficient. Feng Zhang lab, MIT, they developed for detection of a virus, a COVID-19 virus, that means severe acute respiratory syndrome virus. In our case, we also use the same mechanism to lysis the DNA fragment by using like a pregnancy strip or like that. So it is possible. If you know the sequence, unique sequence in the virus, you can target it. Thank you. Tanjina Tuli, how do design gene-specific guide RNA for CRISPR-Cas system? Yeah, it's an interesting question. If you know the specific sequence that you are targeting, gene sequence, then there are some software, even open source software. So in internet you can go there and you can quickly make it, design it, and it is not very complicated. Thank you. Khaled Mahmood Sujon, what is the best tool for designing a guide RNA? Which one is best? I am not sure, because lots of tools are now available. Even lots of companies are now offering the CRISPR-Cas toolkit, guide RNA design and other things. If you know the your cell as well as the sequence you are targeting, sequences even several genes, so you can ask for designing the guide RNA for you. And if you want to do and check for any gene, you can visit the internet, Google. You can browse lots of tools, bioinformatics tools are available there, and lots of examples also are there. And even in the YouTube you can find a very nice video guiding you to design a guide RNA.
So Khaled Mahmoud Sujon, they're like, if you want to do it by home, you can use SnapGene or Benchling, even IDT tools, if you just want to do it right now.
Right.
And after that, you actually have to follow like WhatsApp.
Thank you, Salman, for
right.
Nafisa Shoili, what is a suitable negative selection marker for introduction a genetic construct into the cancer cell line genome? This is a tough question because I'm also curious about the cancer, because in every large family, the prevalence of cancer is now common. And this is a very interesting question, but negative selection marker are very difficult for me to answer, because I have never been tried. But Professor Zakir Hussain, he could answer, because cancer cell, they are really dangerous cell, do not abide by the apoptosis, and problematic for our life, like and give you the perfect answer here. Shah Imran Hussain, how can we use CRISPR-Cas technology for transporter engineering in yeast? Yes, in case of yeast is a model organism, and like Arabidopsis in plant. So in case of yeast, you can easily do engineering, transporter engineering in yeast engineering. If you know the specific sequence which you want to change or edit, or if you want to test a new gene in the yeast, you can do. Even human, insulin biosynthesizing gene have been engineered. Genes are engineered in yeast for making the insulin or like that. So yeast, in case of yeast, it is easier to use the CRISPR-Cas technique. Shagorika Shahriar, do you know something about applying CRISPR-Cas system in Acinetobacter baumannii infections? I didn't work with the Acinetobacter baumannii infections, but Acinetobacter baumannii, this pathogenic bacterium, its genome sequences are available. In case of bacteria, it is very easy because in case of plant cell, due to cell wall transformation is difficult. In case of bacteria, you can, if you can design the guide RNA and CRISPR-Cas system, the construct, you can culture with the bacteria, and you can check the phenotype if you target a specific gene, and you can do it very quickly. Excellent, thank you. Tanjim Hassan, how frequently will homologous recombination happens in E. coli with a red system and a double-strand break? In case of E. coli or any bacteria, it is very easy. E. coli, another widely studied organism, and it is also a model bacterium. So in case of homologous recombination, in case of E. coli, the efficiency should be higher. I am not exactly sure the efficiency rate, but it should be higher, and a double-strand break should be efficient enough. Tanjim Ishraq Rahman, how do electrophorate RNP complexes, nickase, D10A, and 2 single-stranded guide RNA CRISPR-Cas? Very interesting. Actually, receiving lots of lectures, your level is quite up, and I'm really highly impressed. Yes, electroporation in case of animal cell is very good, but in case of plant cell, we can use bombardment technique, as well as we can use gene gun. Even Professor Tofazzal Islam Shiraz, she tried in vivo CRISPR also, like not without tissue culture. So in case of RNP complexes, I think electroporation is efficient, and there are protocols already being established, lots of protocol. And you can, if you need my book, electronic copy, you can send me email. And Tofazzal Islam, this name is unique because Tofazzal Hussain is common. So you can browse Tofazzal Islam and you can find my email, and you can send me, and you can find this protocol in my book. Nigar Sultana, what is the best PCR strategy to genotype large deletion? Large deletion, PCR strategy, the best one. You have to design the primer, and if large deletion, actually sequencing is the best. If big segment is somehow you delete and want to see the what happened, then Illumina sequencing would be the best. But PCR strategy, at certain level, you can design the primer depending on the sequence. Sadrina Afrin Mauna, how can I package 2 plasmid system with lambda red recombination in phase mid to deliver into target cell? It depends on which cell you are targeting. If it is microbacteria like that, then you can easily culture together, and it is easy. But in case of other system, you need, for example, in case of plant, you have to see the multiplex, gene editing protocol, and you design the guide RNA for a particular sequence, and it can be done very easily. Thank you. Jarin Tasnim Rafa, sir, how do deal with non-proliferating fibroblast in single cell culture? It's complicated question because non-proliferative fibroblast in single cell culture, I'm not sure. If non-proliferate, then how can you culture? So I think human biologist can answer this question. I am not familiar. I'm sorry. Upon Islam, is CRISPR-Cas employed in Bangladesh? If yes, then which are the labs working with this technique? Very good question. Java Appa's lab, she told she already applied the CRISPR-Cas. In our case, we started before the coronavirus because our two research team members, they went to UK to work at the laboratory of Sufyan Kamaun for wheat genome editing, and they were successful there. But in Bangladesh, we started the work, but coronavirus disrupted some of our work. But hopefully, next lecture, I shall tell you that we are working on it, because we are preparing in many directions to use it in editing fungi as well as bacteria and plant. Thank you very much for a nice question. And I'm not sure some other lab in other universities or institutes may be using, but I have no information that they have already been successful in using CRISPR-Cas in editing the plant genome or like that. Thank you, Dr. Aparna Islam, for nice question.
Thank you, ma'am.
Marjuk Alam. Good evening, sir. Given the greatly traditional agricultural sector of Bangladesh, what issues, if any, may CRISPR-edited plants face in ultimate commercialization? Also, will the economic aspects be viable for mass implementation? Very nice question. Yes. In case of Bangladesh, I am a member of National Core Committee of Crop Biotechnology, but I raised this question several times. They say if no transgene, no need any law, you can go for the field. But practically, I think we need to introduce some regulation. For the regulation means if a mutant you will get the green pass, we should have some national law. In case of research, we are getting funding from the government for gene editing, and we are going to make a big collaboration with Global Institute of Food Security in Canada, and our two governments, Canada and Bangladesh, already been signed for that one. And I am leading the genomics and phenomics group, and we hopefully would not face a big problem for commercialization of the genome edited, especially non-transgenic CRISPR product, because our agriculture ministry and government is very positive for advanced science, because we introduced already BT brinjal, a transgenic food crop. And your question, another question is viable for mass implementation. I think it is viable. Most important is we need to enhance our capacity. That means human capital, laboratory facilities, and mindset. And obviously, in the curricula, we need to include new content associated with gene editing, so that new generation would lead to make robust use in the society. I forgot to mention Aparna's question. Aparna, I heard Bangladesh Rice Research Institute, they are trying to introduce CRISPR-Cas gene editing for making aromatic rice. We also started the same project, but they are also at the beginning stage, because Dr. Horendra, he received training in China on plant gene editing, and he recently confirmed me that they are at the beginning, at the construction stage. Thank you. Muhammad Rafiqul Islam, great and wonderful talk. Heartiest congratulations for you. Thank you very much for this encouraging comment. I am dedicated to the young people rather than science, because our young generation should be much more smarter and powerful than us if we want to see our nation as a developed one. Sadrina Afrin Mauna, thank you so much, sir, for answering my question from the previous live about the Sherlock technology and SARS-CoV. I have been curious for a while as this is extremely interesting because QRT-PCR has been go to for a really long time before this. Yes, Sherlock, it stands for specific high sensitivity enzymatic reporter unlocking, allows multiplexed, portable, and ultra sensitive detection of RNA or DNA. So Feng Zhang, who was also a very powerful and smartest young guy, and people thought that he would also be included in the Nobel Prize for the discovery of CRISPR-Cas, because they had very big fight for the patent war. Billions of dollars to institutes spent. So Feng Zhang developed the Sherlock method for the very quick detection of the coronavirus, SARS-CoV-2. Thank you. Saeed Anwar, does it mean you are trying to knock out specific genes? Yes, we are trying to knock out. Knockout means we are trying to inactivate, make non-functional S gene in the genome of wheat. And we are successful in collaboration with Sufyan as well as Nick Talbot, because we received funding from received funding from British Biotechnology Research Council. For the young people, I would like to tell you that in 2016, when I proposed that CRISPR-Cas gene editing would be the best approach for development of new blast-resistant wheat variety, at that time, our scientists thought that it is a blue sky. This is why I tried to get funding from the British Biotechnology Research Council in collaboration with Sufyan and Nick, and we did the prove the concept over there. Our target is, once we prove the concept over there, then we can edit for commercial varieties in our country and apply it in the practical field. What was the rest part of the question? Salman, can you show me?
Yeah, actually the question, yeah, right here.
Yes, I guess you need to culture them with enriched, like maybe high FBS or COS or media on polylysine or P-gelatin coated plates. Yes, you are right. We are also using those plates. Thank you.
Yes, I think we got, yeah. So we have all the questions, but if we keep continue, things will be like that. And and I'm sure everyone is enjoying this live. They're having questions, and Sir has been answering questions for a long time, and I'm sure he can go on. And everyone, thank you so much for your participation, and thank you so much for your time. And also, Tofazzal Sir here. We have some observation that the quality of the questions, the way they're curating their questions and coming up with that is pretty amazing. So see how we are here, last couple of years. I mean, maybe 2-3 years ago, things were kind of different from now. But right now, the way people are engaging, and I want to thank people like Tofazzal Sir, and also people like Zara Madam, people like everyone, previous speakers and upcoming speakers. So, and they have been contributing to this society, and they are engaging the students for this learning mediator approaches. So I would like to end this question session today, and also we have 2 minutes to end our live. So I'm gonna thank Tofazzal Sir for your insightful and informative session. And I think this whole thing will be very informative, because I'm sure many of the students, they're gonna watch or rewatch this again and again to extract many information and use that for our own study or research, or maybe sometime motivation. We never know what kind of what kind of information can be used for what. So, sir, we're going to end here, and whoever watching Thank you so much for your time. We are actually honored to be able to organize this kind of event. ধন্যবাদ সবাইকে। স্যার, আপনার যদি কোনো ক্লোজিং রিমার্কস থাকে আপনি আমাদের আজকে ব্যক্ত করতে পারেন।
থ্যাংক ইউ ভেরি মাচ। আসলে এই করোনা একটা আমার মনে হয় মানব জাতির ইতিহাসে এখন আমরা যে এই যে ডিজিটাল এই যে বাস করছি, এটা যেভাবে রেকর্ডেড হচ্ছে করোনার যে ইস্যু এবং এই মহামারী, এইটা হয়তো শত শত বছর পরেও মানুষ স্মরণ করবে। কিন্তু করোনাকে নিয়ে যদি কেউ স্টাডি করে, এটা একটা বড় রকমের কার্স এবং বিগ থ্রেট ফর দ্য হিউম্যান সার্ভাইভাল অন দিস আর্থ। বাট এই করোনার সময়ে কমিউনিটি অফ বায়োটেকনোলজি, ভার্চুয়াল, এটা একটা এক্সাম্পল আমি দিলাম, ভার্চুয়াল অ্যাক্টিভিটি, মানে এই যে একটা অ্যাডভার্স সিচুয়েশনে অ্যাডাপ্ট করে বেরিয়ে আসার যে চেষ্টা, এইটা কিন্তু আমাদেরকে করোনার মধ্যে আমরা কিন্তু প্রমাণ করতে সক্ষম হয়েছি। কাজে কমিউনিটি অফ বায়োটেকনোলজি এইটা একটা এক্সাম্পল, এবং আমার কাছে মনে হচ্ছে যে এই কমিউনিটি অফ বায়োটেকনোলজি যেভাবে ইমার্জ করেছে, এবং এইটার আমি সাস্টেইনেবিলিটি দেখতে পাচ্ছি। কারণ করোনা চলে গেল, এই ভার্চুয়াল অ্যাক্টিভিটিতে যেভাবে আমরা হ্যাবিচুয়েটেড হচ্ছি, এইটা আমাদের ইয়াং জেনারেশনকে, আমাদের এই জেনারেশনে ইভেন বয়স্ক লোকগুলোকেও যেভাবে এনগেজ করেছে ভার্চুয়াল অ্যাক্টিভিটিতে, আমরা কিন্তু আরেকটা জগতের সন্ধান পেয়েছি। আমি একটা জিনিস আপনাদেরকে বলি, এই জগতের সন্ধান এত বিশাল জগৎ, এটা ক্রিসপারের চেয়েও পাওয়ারফুল। এক সেকেন্ডে পৃথিবীর এক মুহূর্ত থেকে আর এক প্রান্ত থেকে আরেক প্রান্তে আমরা ছুটে বেড়াতে পারি। কাজেই এই যে একটা বেরিয়ে আসার জন্য আমাদের বিভিন্ন রকমের ইনোভেটিভ অ্যাপ্রোচ, এবং এই যে আপনার ফ্রাস্ট্রেটেড না হয়ে, আমরা একটা ডেভেলপিং কান্ট্রিতে বাস করি, লটস অফ আপনার নেগেটিভ থিং অ্যারাউন্ড আস, কিন্তু এর থেকে বের হয়ে যে পজিটিভ থিঙ্কিং করা, টু মেক এ চেঞ্জ ইন দ্য সোসাইটি, এবং আদার যে সোসাইটিগুলো যেগুলো দেখে আমরা অনেক সময় চমকে যাই, তাদের চেয়ে যে আমরা বেটার জায়গায় পৌঁছতে পারি, এই যে চেষ্টাটা, এটা করার যে সামর্থ্য আমি ইয়াংদের মধ্যে এই করোনাকালীন সময়ে দেখছি। ইভেন ক্লাসরুমে আমি 3টা কোর্স আপনার অফার করি, একটা পিএইচডি, মাস্টার্স এবং আন্ডারগ্রেড লেভেলে। ইটস অ্যামেজিং। এবং আমার এনার্জি আপনাদেরকে বলি, এই করোনার সময়ে এনার্জি আরো অনেক কয়েক গুণ বেড়ে গেছে, অথচ বাসায় থেকে কিন্তু কমে যাওয়ার কথা। এটার মেইন কারণ হচ্ছে, ফুয়েল হচ্ছে দ্য ইন্টারেকশন উইথ হিউম্যান, স্পেশালি ইয়াং মাইন্ডস ইনক্রেডিবলি বেড়ে গেছে। আমাদের এই করোনাকালীন সময়ে আমার টিম থেকে 30-র মতো রিসার্চ আর্টিকেল ইন্টারন্যাশনাল জার্নালে পাবলিশ হচ্ছে, 2টা বই এডিট করছি। এইটা কিন্তু আমার কাছে মনে হচ্ছে করোনা না থাকাকালীন সময়ে আমার 2 ইয়ার্সের জব। দ্যাট মিনস আমাদের এফিসিয়েন্সি বাড়িয়েছে। কাজেই স্ট্রাগল যেখানেই আছে, এখানে কিন্তু এক্সিস্টেন্সের জন্য এভাবে বেরিয়ে থাকা। আমাদের ইয়াং জেনারেশনদেরকে আমি বলবো যে জগৎটা একেবারেই উন্মুক্ত, বি অ্যাম্বিশাস, এবং যদি ডিটারমিন থাকো তোমার গোলে, তোমার সামনে রাস্তা একেবারে নিশ্চিতভাবে খুলে যাবে, এবং দরজা কিভাবে খুলবে এবং কখন খুলে যাবে এটা তুমি জানো না, কিন্তু তোমাকে 1 বাই 1 নক করতে হবে। কাজেই কখনোই 4টা দরজায় নক করার পরে যদি কোনোটাই না খুলছে, এর মানে ইট ইজ নট দ্য এন্ড। কাজেই আমি আশা করি আমরা যে কাজগুলো করতে পারিনি, যে জগতে আমরা বসবাস করেছিলাম, যেমন আমি বিশেষ করে যারা ইউনিভার্সিটিতে পড়ে বা ইয়াং রিসার্চার আছে, আমি যখন ইভেন ইউনিভার্সিটিতে অ্যাসিস্ট্যান্ট প্রফেসর, তখন হাতে লিখে আমাকে বিদেশে একজন প্রফেসরের কাছে চিঠি লিখতে হয়েছে, এবং আপনার প্রায় 1 মাসের মতো অপেক্ষা করা বা 15 দিনের অপেক্ষা করা যে কখন উত্তর আসবে। কিন্তু এখন জগৎটা একটা ক্লিক অ্যাওয়ে। আমি আমার যে চেষ্টাটা এখন করছি, আমি 10-12 বছর দেশের বাইরে ছিলাম, ইউএসএ তে, ইউকে, জাপান, জার্মানি, ডেভেলপ নেশনগুলোর যে সিক্রেট তাদের ডেভেলপমেন্টের, এইগুলো বোঝার চেষ্টা করেছি। এখন যেটা চেষ্টা করছি, ইয়াংদের জন্য একটা প্ল্যাটফর্ম তৈরি করা, যেখান থেকে গ্লোবালি কম্পিটিটিভ রিসার্চ করা সম্ভব। এই কনফিডেন্সটুকু ইয়াংদেরকে দেয়া, এটা হচ্ছে আমার মেইন কাজ। কিন্তু আমি জানি অনেক প্রবলেম, আমাদের ইয়াংদের অনেক চ্যালেঞ্জ আছে, এই চ্যালেঞ্জগুলো আমি সমাধান করতে পারবো না, কিন্তু আমি একটা জিনিস পারবো, একটা এভিডেন্স তাদের সামনে দেয়া যে উই ক্যান ডু, আমরা পারি। এবং এই কনফিডেন্সটা, এই এক্সাম্পলটা যদি আমাদের নতুন জেনারেশনকে উদ্বুদ্ধ করে, তখন আমার কাছে মনে হয় এটা হবে আমার জন্য সবচেয়ে বড় রকমের অনুপ্রেরণা। আমি সবার সুস্বাস্থ্য কামনা করছি, সবাই নিরাপদ থাকেন, কারণ এই কোভিড-19 এটা এক্স্যাক্টলি কবে শেষ হবে উই ডোন্ট নো, এবং শীতে বলা হচ্ছে যে এটা প্রকোপ আরো বাড়তে পারে। সবাই সাবধান থাকবেন, স্বাস্থ্য অভ্যাস মেনে চলবেন, আর নিজের যে স্বপ্নটা আছে এই স্বপ্নটাকে খুব ফোকাস করে ওই দিকে ছুটবেন। জীবন মানেই আসলে এইভাবে মহৎ স্বপ্নকে নিয়ে ছুটে বেড়ানো, এবং এটা আমাদের লাইফ স্প্যান খুবই শর্ট, সময়কে আমরা প্রত্যেকেই মর্যাদা দিব। এবং আমি খুবই খুবই ইমপ্রেসড, বিশেষ করে কমিউনিটি অফ বায়োটেকনোলজি, সালমান আছে, নাইরিতা আছে, ইউসা আরাফ, অ্যামেজিং পিপল। আমরা কিন্তু আমাদের স্টুডেন্ট লাইফে আমরা এইরকম চমক দেখাতে পারিনি। আমি গ্যারান্টি দিয়া বলতে পারি তারা ওয়ার্ল্ড লেভেলে তাদের যে স্মার্টনেস, তাদের যে ডেডিকেশন এবং প্যাশন টু লার্ন নিউ থিংস, এবং তারা যেভাবে সোসাইটিকে নিয়ে চিন্তা করে, একটা কমিউনিটি অফ বায়োটেকনোলজি, এটা হচ্ছে একটা সোসাইটিকে শেক দিয়ে সামনের দিকে নিয়ে আসার, এটা একটা রিভলিউশনারি অ্যাপ্রোচ। কাজে আমাদের দিক থেকে, আমার দিক থেকে 100% সাপোর্ট থাকবে। আমি নিজেকে ধন্য মনে করব এই ধরনের মিশনকে এগিয়ে নিয়ে যাওয়ার জন্য। আমি তোমাদেরকে এইটুক গ্যারান্টি দিতে পারি যে আমাদের মতো যারা সিনিয়র আছে, ইভেন আমাদের চেয়ে অনেক সিনিয়র যারা আছে, ইভেন জেবাপার ফাদার আহমেদ শামসুল ইসলাম, উনি 60-এর দশকে বাংলাদেশে বসে নেচারে আপনার 2টো আর্টিকেল পাবলিশ করেছিল। আমাকে ইন্সপায়ার করে কিন্তু ওই সমস্ত শক্তিগুলো, যে আমরা আসলেই কিন্তু এরকম ক্যাপেবল। আমি কখনোই কিন্তু মনে করি না বাংলাদেশে বসে রিসার্চ করার কারণে আমরা কোনো দিক থেকে গ্লোবালি সোশ্যালি কোনো ডিসক্রিমিনেশনের শিকার হতে পারবো। বি কনফিডেন্ট ইউরসেলফ, এবং যদি নিজের পাওয়ার থাকে, জগৎ কিন্তু স্বীকৃতি দেওয়ার জন্য রেডি হয়ে আছে। আমি যে বয়সে বিজ্ঞান একাডেমির ফেলো হয়েছে আমি নিজ স্বপ্ন কল্পনা করিনি। এই বয়সে কিন্তু ইনফ্যাক্ট ফেলো হওয়ার কথা না, কারণ এর আগে 60-এর উপরে যারা গেছে তারা ফেলো হয়েছে। এর মানে সোসাইটি ইজ রেডি টু কনগ্রেচুলেট ইউ। কাজে আমরা আমাদের আমি মনে করি শুধুমাত্র সেলফিশলি না, এই যে কমিউনিটির মাধ্যমে এগিয়ে যাওয়া, একটা সোসাইটিকে পরিবর্তন করতে হলে এই কনসার্টেড এফোর্টটা খুব ইম্পর্টেন্ট। এবং আমার একটা বড় শক্তি আছে, আমি এটা বলে শেষ করি আমার বক্তব্য। সেটা হচ্ছে, যদি কেউ আমার পেপারগুলো কেউ দেখে, মনে করো যে তুমি অ্যারাউন্ড 200 পেপার আছে, তুমি মনে হয় পৃথিবীতে কোনো কান্ট্রি খুঁজে পাবা না যে কান্ট্রির লোক কুয়াতার নাই। এবং আরেকটা জিনিস খুব ইন্টারেস্টিং দেখবা, আমি এটা খুব এনজয় করি, অলওয়েজ নিউ জার্নালে পাবলিশ করা। মেজরিটি ক্লাসিক্যাল রিসার্চার দেখবা একই জার্নালে সিরিজ অফ পেপার পাবলিশ করে। আমাদের এই রিসেন্ট পেপারটা যে দেখছো, যে ক্রিসপার যে স্ট্রিপ আমরা তৈরি করেছি, এটা হচ্ছে ইঞ্জিনিয়ারিং জার্নাল। এটাতে কিন্তু নাসার পেপারও পাবলিশ করে, পদ্মা সেতুর পেপারও পাবলিশ করে। ইঞ্জিনিয়ারিং মানে হচ্ছে বাউন্ডলেস। আমরা বায়োলজিক্যাল ইঞ্জিনিয়ারিং রেজাল্ট পাবলিশ করেছি। দিস ইজ এ কিউরিওসিটি, এবং আমি এই কিউরিওসিটি গুলো মেনটেন করি। পৃথিবীর যত বড় ব্যক্তি হোক তাকে কিন্তু নক করে। এবং তোমাদেরকে আজকে একটা শেয়ার করি, কয়েকদিন আগে একটা লেকচার দিয়েছিল জেনিফার ডাউডনা, আফটার এই নোবেল প্রাইজ উইন করার পরে সিনথেটিকো তারা অর্গানাইজ করেছিল ক্রিসপার ডেতে, ওয়ার্ল্ড ক্রিসপার ডে। ওখানে থাউজেন্ডস অফ কোশ্চেন হয়েছে। তা আমি একটা কোশ্চেন করেছিলাম, ফরচুনেটলি শি আনসার্ড দ্য কোশ্চেন। এবং কোশ্চেনটা ছিল কি, যে আমরা এই যে প্লান্টের রুট রাইজোস্ফিয়ারে যে মিলিয়নস অফ মাইক্রো অর্গানিজম আছে, যারা প্ল্যান্টের গ্রোথ, ভাইটালিটি এবং সমস্ত কিছুকে রেগুলেট করে, প্রোবায়োটিক। যেমন আমাদের হিউম্যানের মধ্যে 3 to 6 পাউন্ড মাইক্রো অর্গানিজম আছে, প্রোবায়োটিক আছে 150 স্পেসিসের মতো। অবাক হয়ে যাবে, 33% আওয়ার জিন হচ্ছে এই প্রোবায়োটিক থেকে এসেছে। এইটাকে জিন এডিটিং এ আমরা বড় রকমের ব্রেক থ্রু আনতে পারি কিনা। উনি বলছে যে এইটা একটা অত্যন্ত ফ্যাসিনেটিং এরিয়া, আমাদের টেকনোলজিক্যাল আরেকটু অ্যাডভান্সমেন্ট হলে আমরা ওখানে ব্রেক থ্রু আনতে পারবো, এবং তখন এগ্রিকালচারটা অন্যরকম এগ্রিকালচার হবে, ওই ক্লাইমেট চেঞ্জ তখন কোনো প্রবলেম হবে না। তা আমি সবাইকে অসংখ্য ধন্যবাদ জানাচ্ছি, বিশেষ করে যারা আমাকে ইনভাইট করেছে। ইট ইজ এ বিগ অনার ফর মি, এবং এই সন্ধ্যাটা আমার চিরস্মরণীয় হয়ে থাকবে। এবং সবাই অনেক ভালো থাকেন, এবং বিশেষ করে এই কমিউনিটি অফ বায়োটেকনোলজি, এটা আমি চাই যে এটা লং লাস্টিং হোক এবং এটা সাস্টেইনেবল হোক। এটার জন্য যে কোনো ধরনের ইভেন ফাইন্যান্সিয়াল সাপোর্টও যদি আমার লাগে, আই উইল বি উইথ ইউ। থ্যাংক ইউ সো মাচ।
Thank you so much, sir. Thank you so much for being such an inspiration and like supporter for this kind of community. And I think with your type of direction, we can grow more. And also, I can see, I can just, resist to say that like many of the students, many of them are actually my students, and I'm like really feeling so proud that they are like putting themselves into this line and they're engaging themselves like that. And also আপনারা সবাইকে ধন্যবাদ জানিয়ে আজকে শেষ করব। ইতিমধ্যে আপনারা দেখতেই পাচ্ছেন যে স্যারের এই উৎসাহী কিংবা এই অংশগ্রহণ সবকিছুর মধ্যে শুধুমাত্র যে বায়োলজির মধ্যে থেমেছে ব্যাপারটা কিন্তু তা নয়। আর এবং বায়োটেকনোলজি ব্যাপারটাই কিন্তু আপনারা এখানে কিন্তু আমরা শুধুমাত্র বায়োলজি বলতে বায়োটেকনোলজি কিন্তু আসছে না। এখানে ইঞ্জিনিয়ারিং থেকে শুরু করে কম্পিউটার সায়েন্স, মাল্টিডিসিপ্লিনারি বিজ্ঞানের কিন্তু বড় একটা মানে ইটস কাইন্ড অফ লাইক আ মেল্টিং পট, রাইট? সো আপনারা স্যারের কথাবার্তা থেকে কিন্তু অলরেডি একটু আভাস পেয়ে গেছেন কিংবা পুরোপুরি জানেন যে যারা আপনারা অলরেডি এই লাইনে আছেন তাদের তো অবশ্যই জানেন, কিন্তু যারা নতুন, যারা এই শোতে আজকে দেখছেন, এই ইভেন্টে আজকে যারা অংশগ্রহণ করেছেন, তারা হয়তো বুঝতেই পারছেন যে বায়োটেকনোলজির মানে পরিসীমা কিন্তু বলতে গেলে নেই। এখানে কিন্তু আমরা যেহেতু জীবনের সাথে আমরা মানে যা সম্ভব, যেটা আমাদের জীবনকে সমৃদ্ধ করতে পারে যে কোনো ভাবেই হোক, সেটা আমাদের চিকিৎসা হোক, খাবার হোক, সেটা আমাদের নতুন নতুন রিসার্চ আবিষ্কার যাই হোক, সবকিছু কিন্তু এর মধ্যে নিবদ্ধ। তথাপি আমরা বলতে পারি বায়োটেকনোলজি সীমাবদ্ধতা আমরা নিজেরাই সময়ের সাথে পার করে ফেলবো। এবং স্যার এখানে আছেন এবং আমাদের যে অংশগ্রহণকারীর মধ্যে অনেকেই আছেন যারা বড় বড় গবেষক, এবং অনেকেই আছে যারা শিক্ষক, অনেকে আছে যারা ছাত্র কিংবা ছাত্রী। তো এই যে সবাইকে মিলে এই কমিউনিটি, তাদের পথ চলাটা আরো সুগম হবে স্যারদের মতো মানুষদের হাত ধরে। এবং আপনাদের সবাইকে শুভেচ্ছা জানিয়ে এবং ধন্যবাদ জানিয়ে আজকে আমাদের লাইভটি শেষ করছি। থ্যাংক ইউ সো মাচ। ধন্যবাদ সবাইকে। শুভরাত্রি।
ধন্যবাদ।